Thanks a lot for the invitation. In this presentation, I'm going to assume, I know that you have very different backgrounds, so I'm going to assume that you have some basic knowledge of data science. I'm trying to explain how this can improve cognitive neuroscience. So cognitive neuroscience addresses the questions of how our cognitive activities are affected or controlled by neural circuits in the brain. Of course, I'm giving here a schematic definition. There are cognitive neuroscientists in this room, so don't kill me after the conference. I know I'm simplifying a little bit. So for those of you who don't know cognitive neuroscience, I think it's useful to give a little bit of historical context. So a long time ago in antiquity, we actually, in the time of Aristotle, for instance, it was thought that the thought came from the heart and that the brain was more a cooling system. So at the 2nd century after Christ, it was first noted that the brain was believed to be the source of mental activity. And then, of course, a lot of time was spent with very slow progress. And I think the last 2 centuries have been the centuries of improvement in our understanding of the brain. So at the beginning of the 19th century, the main field was, the main stream was phrenology, where it was thought that there were about 30 areas in the brain, and if you could look at the shape of the skull, you could make inference about the ability of a person. So this is the idea of Bose-Demat, where there is a certain region which makes you good at mathematics, and if we can find this, we could make inference about your cognitive abilities. This theory was a bit weak in terms of scientific evidence and was then abandoned. Then there was the aggregate view on brain function, which was based on the observation that actually when you do some cognitive task, many brain systems are involved in it. So the cerebellum, the cortex, subcortical structures are actually jointly involved when you perform a certain cognitive task. And so the idea that everything was working together. But then later come some evidence from lesions that people with lesions in some part of the brain, in what is known as Broca's area, Wernicke's area, had very selective disabilities, like in language understanding or production. And this gave a more localizationist point of view, where actually some specific brain region would be specifically involved in some cognitive task. And actually, the aggregate view was abandoned, more or less. And this localizationist view was more prevalent then. Actually, the truth is a bit more complex. And we see that many systems are actually involved in cognition. But it is also true that there is some specialization in the brain territories. Then there are lots of side discoveries. I think cognitive neuroscience is at the convergence of many fields, so biology and neuroscience. For instance, it was observed at the end of the 19th century that blood flow is associated with brain function. And this is great because it's very hard to measure brain function, but it can be easier to measure blood flow. And this is what is used in current brain imaging techniques. Then there was very impressive progress at the beginning of the 20th century on the analysis of neurons. A long time ago, people didn't even know how the nervous tissue was built. So it was considered like a kind of continuous tissue. But actually, it was discovered that it was made of neurons, which are single entities, which could be treated like agents that exchange information with synapses. And so we know that we have about 100 billion neurons in the brain and about 10,000 synapses per neuron. And then more on the side of cognitive theories, there was a cognitive revolution, which was that the Pavlovian approach, where you've got a stimulus followed by a response, which was prevalent at that time, was not complex enough to represent the behavior, the complex behavior. For instance, our understanding of language, our more abstract abilities. And so more complex theories were developed, and this was a cognitive revolution. And then, of course, there was huge progress along the 20th century. Many of them based on lesion studies, so people with stroke with a very focal lesion. And we could study, for instance, the impact of lesion, or people with epilepsy. So I'm skipping a lot of steps, but I think a very important one is with Hubel and Wiesel, who worked on the visual cortex of cats and provide a very simple explanation in some way of how these neurons were responding. So essentially, neurons in the primary visual cortex responded as edge detector, like filters that respond to sharp contrast in the input images, oriented contrast. And then this was further developed, and I mean, the theory of this was developed by David Marr, for instance, in his work on visions. I think a very important step then was in the 90s, the use of non-invasive imaging, like functional magnetic resonance imaging and magnetoencephalography, so fMRI and MEG, that can be used in humans. So non-invasively, we can hire anybody and ask them to do an experiment and look at how their brain responds to some stimuli, which of course makes it much easier to run experiments. And it's still currently the main source of data on at least on human cognition. There's a completely different field of animal imaging with electrophysiological recordings, etc. So I'm not talking about that today. And maybe another revolution which is still ongoing is optogenetics. You don't really see it, but it's optogenetics, where we can directly manipulate the activity of neurons, so in animals. I think that what I would like to keep from this brief perspective is that we've been limited, and we're still limited by measurements. We're not, I think, limited by theory to understand the brain. We're limited by measurements and by data. So the way I see human cognitive neuroscience today is the following one. People build theories on how the brain solves different problems, like how do we model consciousness, for instance, in the brain, or how do we model language processes in the brain. And to probe these theories, these hypotheses, we build experiments that involve some stimuli, and they are organized in what we call an experimental paradigm. So a sequence of operations done by a subject, so which can be done just behaviorally outside of a scanner. But now we can take also a living brain, which is in a person, put the person in the scanner, and find some way to present the stimuli to this person with some visual devices or auditory devices. And then the person will do some actions. And then we analyze the response to these actions. And this provides these brain images with these colored blobs that represent how different brain regions respond to the stimuli. In terms of data analysis, we will try to make the link between the presented stimuli and the activation data. So in one direction, we could call this an encoding problem. So how the activity associated with the stimuli are encoded into brain activity patterns. Or you could see that as a decoding problem, where you, based on brain activity, you would like to make some inference of the occurrence of a certain cognitive activity related to a stimulus. So in this talk, I will be talking only about functional magnetic resonance imaging, fMRI. So fMRI can be obtained in MRI scanners fairly easily. It's a non-invasive modality. So assume that the subject, the participant, views a visual flash at time t equals 0, and that you are sitting here in the occipital cortex and observing the signal. So ideally, if you had perfect observations, you would see a slow increase of the MR signal that takes 5-6 seconds, and then a decrease, an undershoot, and a return to baseline in about 20 seconds. This signal increase corresponds to the arrival of oxygenated blood, which is used to compensate the consumption of oxygen during neural activity. But it's an overcompensation, so actually the concentration of oxygenated blood increases, which creates this slow but fairly robust signal increase here. Of course, we're not a perfect observer. We can only sample this signal discretely. And what happens with MRI is that you can take pictures of the brain about every 2 seconds. So every 2 seconds, you have a complete 3D volume of the brain with a resolution of currently 1.5 millimeters. So current technology make it possible to scan the full brain in with a resolution of 1.5 millimeter in 2 seconds. It's great. I think it's among the non-invasive modalities, it's maybe the one with the best resolution, but it's not good enough. I mean, in 1.5 cubic millimeter voxel, you have about 10,000 neurons, maybe several tens of thousands of neurons. A voxel, sorry, is a volumic pixel. So when you have volumic images, you call the volume elements voxels. And so what you have at the end is for each location a discrete sampling of this time response to the stimulus, which is a very slow response, much slower than the dynamics of brain activity. And with this, you can vary the conditions that you use to map the brain and look at the response to many different conditions. For instance, you can ask the subject to move the right hand, actually to press a button with the right hand or the left hand. And if you do the subtraction, what is the activity for right hand button press versus left hand button press, you see this blob in the left motor cortex, which is the region that controls the hand motion, actually even in the S2 region here in this area. But you can also do more complex experiments. For instance, in this experiment, the subject is listening to stories. Some of them, so the subject has to figure out the explanation for a certain very short story. Some explanations can be completely mechanistic, or some of them might believe a manipulation of the belief of others. So for instance, somebody wrongly believes that, I don't know, the other has done something nasty to him or to her, etc. So when you, and then we compare the brain activity during this false belief task with the brain activity during the mechanistic task. And then we see that in the false belief condition, you will have this specific activity here in the parietal lobar region or in the STS. And this is part of what we call the social brain, etc. So a good cognitive neuroscientist is somebody who can craft experiments that are good at specifically targeting some brain systems and with very high-level controls, which mean that you can isolate fairly accurately some brain processes.
How many times do you have to repeat the experiment to average these?
Typically, you would do 10, 20, 30 trials, essentially, to have a high SNR. The more, the better. But then you have to do some compromise with cost of scanning time, etc., and subject tiredness, etc.
The color code, please. Is it only the intensity of the...
So these are statistical maps that show the effects. So essentially, this represents the effects that are larger than 3 times their standard deviation. So, and then of course, the more yellowish, the stronger the effect.
The blue, for example, what is that?
Oh, the blue is a deactivation, yes, sorry, because you can have the positive effect or the negative effect. Since we're always doing subtractions, actually, the subtractive reasoning is really at the basis of this way of looking at the brain. We compare the activity in condition A versus B, and we assume that the only difference between A and B is the cognitive process that we try to isolate, which is wrong, of course. Another point of view, also on brain imaging, it's maybe brain imaging as a medical imaging modality and a useful one, which is good at getting some biomarkers, some signals to compare populations. For instance, in psychiatry, to study some brain diseases, we still don't understand well the many brain diseases, and we don't even have the right categories. We don't know what an autistic kid is, because there are several diseases that are called autism, and we don't, at the moment, we don't understand them. So understanding them will require lots of effort in genetics, but also brain imaging can help clarifying what makes these kids different and whether there are several subgroups among the population. So for that, we use brain imaging to extract some biomarkers and compare populations. Actually, one great thing is that you don't even need to do a task to do this kind of thing. You can perfectly acquire data while the subject is resting in the scanner and simply compute the covariance or the correlation structure of the brain signal. We know that this correlation structure is perturbed, is affected by diseases, and by doing some machine learning on these covariance matrices, we can make some inference on the state of the subject. We can make some diagnosis. Okay, so far I've described the field. Now let's go to the problems. Of course, the first problem is that the images are noisy because we never have enough trials, we never have enough observations to have a high enough SNR. The second one is specific to the human brain, I think, is that there's a very impressive variability in the structure of the brain. There's not even a real standard brain. We don't have a standard shape to represent brains. We can only approximately match brains to templates. Then, when you look across experiments, you've got a labeling problem. For instance, 2 minutes ago, I talked about the false belief experiment, etc. Well, there are many different ways to describe this experiment. I use some terms, but the description I've given are partial, and another lab would probably give a different description of similar material. And so, when you think in terms of knowledge accumulation, how do you relate brain to cognitive categories, it's really a problem of labeling, which has not been very well described so far, but I think is very important for the future. And finally, images are high dimensional, and this is more a statistical problem that we will call curse of dimensionality, multiple comparison problems, and sensitivity issues when we do statistical inference. I think many of these points are common to many different fields of data science, so I won't discuss all of them. Just illustrate here one of them, which is between subject variability in brain activity. So here you have 5 different people, so each column is a different person that have done 4 different tasks. So each row is a different task. So I've taken the same, in a standard space, I've taken a slice in the same position to be able to compare people. And of course, you see some similarity here. The person is doing a motor task, and so you can see the motor cortex activated. Here it's also a motor task, I think. This one is a comparison between auditory and visual activity. So you see the auditory cortex in red colors and the visual in blue colors. And here it's a task that involves mental arithmetics. The subjects do subtractions. And so you can see that there is, of course, some commonalities between some subjects, but also very huge differences in the level of signal, in the SNR per data set. So in some cases, the activations are fairly weak. In the detail of the shape of the pattern, you can see some differences, and sometimes in the large-scale organization of the patterns. Yes?
For example, we talked about motor cortex. Is the localization of the motor cortex the same from one individual to the other? What could be the explanation that the region is displaced from subject?
Yeah, so, yeah.
should try to correct for that?
Yeah, no, absolutely. The community has been working on that for a long time. So we know that the motor cortex has essentially the same position at the macroscopic scale for everybody. It's just before the central sulcus, which is one of the main folds of the brain. So it's easy to find. Now, if you look at the details, of course, it's very hard to take even the central sulcus of person A and match it to the central sulcus of person B, because these sulci don't have exactly the same shape across people. And so you need a lot of information to be able to match subject A with subject B. And the motor cortex, I would say, is the easiest one, maybe also the visual cortex. But for other systems, it becomes a big mess. If you take all the frontal lobe, there's not even a shape consistency between people. Okay, now brain imaging. Is brain imaging in the big data era? Not really. I would say that neuroscientists don't like big data. For them, it's not the way they work. You know, when science is done by PhD students, and quite often a PhD student in cognitive neuroscience is interested by a question, so he will try to craft a paradigm, to test some hypotheses, discuss this hypothesis, build all the material to run the experiment, analyze the data, which is time-consuming and difficult, and then maybe do more experiments because you don't have the effects that you expected, and then finally reanalyze, potentially publish a paper, etc. When this is done, the PhD thesis is over, and it's the next student who comes. The data are lost quite often, and so it's very hard to accumulate data and to have a big data approach in this context. The labs do not commonly share their data, and so actually, currently, people have not accumulated data. And actually, since it's time-consuming and difficult to have large cohorts of subjects, people have even invented arguments to say we should not sample large populations. And people have said that n equals 16 subjects was the ideal cohort size for brain mapping, which is completely wrong. Because when you try to detect effect in small populations, you end up with reproducibility issues where by chance you observed an effect in a small population, or you tortured the data strongly enough to get the effect you wanted, but this will never generalize to a larger population. And people have recently realized that with this approach, you could not have a reproducible cognitive neuroscience. So in this talk, I'm going to discuss some attempts that we've done to analyze larger data sets and improve statistical inference based on larger data sets and also various data. And then I will discuss more technical aspects on how do we handle large data sets. And finally, I would like to discuss some work on the relationships between artificial intelligence and brain modeling. So large-scale analysis of brain function means using predictive model to build an accurate model of brain function. For this, we will have we can study it as a forward model, which are called encoding, in which you present stimuli and look at the effect of the stimuli. Now the problem is, when you look at a place image, a tool image, or a checkerboard, you can see this activity in the primary visual cortex. So what does this activity mean? Can we decode this activity? And we cannot answer this question with this forward mapping. To answer this question, we need to go the other way, the way up, where we try to predict which stimulus was presented given the activity. And so can we predict whether the subject was attending to a checkerboard, a tool, a place, or moving the hand, for instance? Once again, historically in the literature, people have only used the forward way. They were getting data, analyzing data, so getting these images with these blobs that I've showed, and then they were just registering the position of the main foci of activity in databases. So you just register a few coordinates, and this summarizes a whole brain scan. And of course, you've got also the problem of describing accurately the experiment that was performed. This is why I call this a broken pipeline. So what we've tried to do instead was to rely on actual data. So we have taken about more than 30 studies comprising about 1,000 subjects, and this represents 15K images, which represent different protocols that have been run in different labs. Some of them are available publicly, and some of them were provided to us by colleagues. And then we try to run forward inference. Forward inference in that case means you try to represent all the experimental conditions that were presented to the subjects in a common vocabulary, an ontology if you want. And so you encode this. This is a design matrix that you can see here. It's just an occurrence matrix that tells you that when you do, for instance, tone counting, this involves auditory activity and counting activity. So you create this big design matrix, and then you use it to explain the brain images by finding the effect betas that explain, that relate to the effect of the term. The problem that comes with this approach is that if you look at the correlation structure in this design matrix, some of the terms are very correlated. For instance, if you take saccades and tracking, these terms go together. When you have a saccade task, this involves tracking with the eye, so probably you won't be able to separate the effect of these two terms using this approach. Similar with counting and digit. Or you've got some anti-correlated terms like auditory, so visual and auditory, for instance, because typically you present a stimulus visually or auditorily, but not both. So you've got some exclusion between these two terms, which creates anti-correlation in the design. So the consequence is that if you look at the effect of the term auditory across all these studies, of course you find the auditory cortex quite well, but you find in negative, in blue, the visual cortex because of the anti-correlation between the two terms. If you look at the term counting, you find some frontal and parietal region that are involved in this task, of course, but you also find visual activity that has not been well separated from the counting task itself, etc. For instance, these are the activation maps associated with two different kinds of visual stimuli. One is abstract patterns, and the other one is shapes. And typically, it's hard to distinguish between these two. So you see the limitation of the forward model that does not discriminate actually between the conditions. So to do that, we want to do the reverse inference. Yeah.
So the image are different stacks, different slices.
Oh yeah. So, sorry. Each image, so it's a full volume, so it's a bit hard to represent. So what I do typically is to select some slices. So these are 6 different slices of an image. So the name of the game now is you give me a brain image, and I would like to try to guess what conditions was associated with this image. So in terms of classification, it's a multi-label classification, not multi-class, because actually you want to associate terms with an image, but the terms are not mutually exclusive. It can be, for instance, a tone counting task, which will involve auditory activity and a counting activity. So now we go from the images to the design matrix. There's a pitfall here. Depending on the way you do your cross-validation, so the way you look at the accuracy of the model, because typically if you've got a tone counting task, only in one study, it's fairly easy to say, oh, this image from this study is very close to the image of the same study. So this is what a classifier does, basically. And even if the classifier does not learn what tone counting means in terms of cognitive representation, just by looking at the similarity between images, there is a study effect which is very large and which can essentially be leveraged by the classifier. So this is given by the distance histograms here. If you look at the average distances between images, so two images, any two images follow this histogram. Two images that share one cognitive label in common follow essentially the same histogram, but two images from the same study, even if they do not correspond to the same condition, have a more shifted histogram, so they are much similar to each other. So if you're a bit awkward, you can simply do something which is like a scanner detector or a study detector, but not something that actually captures the cognitive processes that underlie the activity. So you need to do a cross-study, cross-validation. So to solve this problem, we have used this complex machine. It's classifiers with 3 layers. It's almost deep learning. It's just a standard classifier, like a logistic regression, but we create an intermediate hidden layer. Essentially, it's interesting. So we put all the images as input with their labels. And the intermediate layer is to try to kind of clustering of the labels that follows some cognitive ontology. And we try to see whether some groups of labels are jointly present or not. And so we create an intermediate representation that discriminates well clusters of labels between images. And then based on this latent representation, we do the final classification. This is this wiring here. And then based on this, we can predict whether each label can or not be applied to brain images. But well, this machinery is more or less impressive. I think the important part here is that we do the cross-validation in leave one study out. And even we've tried leave one lab out, because the data from one lab look like the data from this lab, and this lab is always interested in language, so it's typically easy to associate an image with language just because it comes from a given scanner or a given lab. So when we do that, if we look at the performance of the prediction of the different cognitive terms, we see that terms like visual, auditory will be well predicted. So these bars represent the precision and recall for the different for the prediction of the different terms. In red, you've got here the chance level obtained by non-parametric means. And in green, you obtain the score that you would have if you were doing leave one subject out instead of leave one study out. Of course, you would have an optimistic bias. The performance would always be higher if you have leave one subject out, but it's for bad reasons, probably. So most terms are decoded much better than chance. Just for instance, visual digits is not decoded at all, because I think it's confounded. There's a confusion between digit and letters, and so far we did not have enough data to see the particular representation of digits as opposed to, for instance, letters, visual letters, etc. Faces are very easy to characterize in terms of brain activity or seeing a place image, but more abstract concepts like inhibition or failed inhibition or gain or loss in gambling tasks are much easier to decode than the more low-level stimuli. So an important thing now is if we look in terms of brain patterns, because, okay, accuracy is nice, but it's not very interesting for cognitive neuroscience. If you take the term place, place here means viewing a place image, like a landscape. If you look at the activity, brain activity associated with it, like in the forward model, you would have all the visual cortex. Actually here, the ventral part of the visual cortex that is activated when you look at a place image. Now, if you try to do a place versus a tool image, or versus a face image, or versus a word image, etc., you see a slightly more specific pattern with 2 spots here, but still some activity in the ventral visual cortex. Now, if you implement a classifier that classify place images versus all other types of stimuli, you end up having a much more focal response with these 2 regions that pop up, which are the parahippocampal place areas. So we decided to take a kind of intersection between these 2, and so we retain this as a pattern. So this summarizes our segmentation. So with that, we obtain an atlas. Sorry, it's a bit dark. It's a bit hard to see. But you see about 10 visual categories that are mapped to the visual cortex, especially the ventral part of the visual cortex. Some motor activities related to feet, hands, and tongue. Some language activities and auditory activities, so related to the listening to sounds or to human voices or to linguistic content. And some activities related to mental arithmetics and saccades. So this summarizes essentially what we can infer from the currently available data in terms of brain activity localization. Yeah?
When you choose your inputs, you can have variations, for example, to take the same individual you are doing measure and show different stimuli. Or you can also have the same stimulus to different people.
Yeah.
So the data you are showing is combination of the two? Or?
Yeah, it's an average model. So it's a consensus model. And as you say, you have several layers of variability. There's a between subject variability, but also a between study variability. When you say, I presented a face image to the person, well, it depends on the image. If it's a natural image, there's some clutter in the image, or if it's on a neutral background, or etc. And currently, we have no way to distinguish between that. So we just try to see whether across all these conditions, we can see a region that robustly predicts the occurrence of viewing a face across all these settings. Okay, I'm a bit slow. I try to hurry up a little bit. The second point now will be, with this, we're coming to rather heavy computation. So how do we deal with large-scale analysis of brain images? So I've already talked about resting state fMRI, which is used currently to study brain structure. So the subject is just resting in the scanner, as you may be in your seat. Maybe nobody's sleeping so far, but even during rest, you can see that there is some activity with some structure. And we can study this structure. It tells us something about the way the brain is wired, because regions that are wired will typically activate together, etc. So we can use this to study the fundamental organization of brain into networks, for instance. And for this, we will take the input images and stack them in a matrix X. So in the matrix, we take all the voxels, we enumerate all the voxels in the volume, and we look them across time, and this creates a data matrix. And a way to analyze it is to decompose it into spatial temporal modes. So the spatial modes will be spatial maps, and temporal modes will be the associated time courses. In a resting state, we don't care about the time courses because, well, they represent some random signal. But we care about the spatial maps because they represent the main networks of the brain. So typically, to obtain this decomposition and to be able to identify the spatial components, you impose some sparsity on the spatial maps, for instance, with an L1 num. So it's a matrix factorization into spatial temporal modes with sparsity in the spatial domain. So it's known as dictionary learning or sparse principal components analysis. Okay, but now if you do that, for instance, you have several hundreds of images per subject. And so the number of observations that you have, if you stack several subjects together, you start to have a rather decent number of samples in this analysis. And the number of voxels is typically 200,000 if you look at the brain with a resolution of 2 millimeters. So the data matrix becomes quite huge. And I like quoting this email I received about 1 year ago. It was on the mailing list of the domain, where people complain that they cannot do this kind of analysis on more than 10 subjects because this blows up the memory of their PC. And the data set comprises 500 subjects. So how do we do? So if we take this data set, we have about, so we have 4,000 volumes per subject and 500 subjects. So this makes 2 million samples. And each sample is 200,000 voxels. So it's a huge matrix. It's 2 terabytes of data. So it will never fit on the memory of your computer. So for that, you need to have some online learning that takes some batch of data, update the model, and then reiterate this procedure. So for the specialist, you know that Julien Méral, for instance, has provided some algorithms to do this online factorization. But still, it's super slow on this data. It will take days of computation to run this. So to be able to be even faster, now what we do is that we are online in the sample. So we take randomly some batch of images to update the model. But we are also online in the feature dimension. We don't update the model for all the voxels of the brain. We just take a batch of voxels and update the model. So the old way, the textbook way to solve the problems is to factorize the whole matrix X. But it's not doable in practice. So a better solution is to stream the data in the sample direction. So you solve the dictionary learning equations, which are just alternated least square, where you basically optimize on the dictionary and on the loadings of the model with some penalties or some constraints that are used to enforce structure in the solution. And so Julien Méral provided 6 years ago some very elegant solutions to this problem. But the next step that we have introduced was to now subsample also in the other dimensions. I'm sorry, the image is cut, but essentially you only update the model for a fraction of the voxels. So what we've done recently was to show that empirically this works, and this can bring huge time savings. And with Julien Méral, actually, we have proved that this is a consistent estimator that will return the right solution to the problem, to the estimation problem. So in practice, for instance, if you were doing this matrix factorization without the undersampling in the feature dimension, you would get the solution in about 10 hours. Now, with the subsampling approach that we have introduced, we can obtain the same result in about 1 hour. So essentially, the ratio of subsampling that you use in the data is the ratio of time you save in the estimation of the model. And of course, what I'm describing here has nothing to do with brain imaging. It's matrix factorization. So you can apply this to any matrix factorization problem, like for multispectral imaging or problems like that. We have, for instance, applied it to recommender systems. And for very large data sets, this brings the same time savings. So with this dictionary learning approach, we can obtain, so from our 2 terabytes of data, we can obtain this nice dictionary of brain patterns, which are regions that represent some modes of activity, of spontaneous activity. So this is the atlas with all the regions put together, and we can delineate all of them. You can see in the details the symmetry of the components, which is very promising. Now, we are in a strange situation because on the one hand, we have huge dataset, resting state datasets, which can be used to learn structure on the brain, but they are unlabeled. So this tells us nothing about cognition. And on the other hand, we have some labeled data, but very scarce data, very few data points. So could we leverage the unlabeled data to improve the learning of components for labeled data, so for cognitive tasks? So that's what we are doing. So it's called semi-supervised learning, where essentially we have 2 models. We have a model which is called an autoencoder, and then tries to create some intermediate representations that are good at reconstructing data here. And we have coupled with it a discriminative model that creates some latent representations of the data, and from this try to classify the data into different tasks, into different labels. So it's done with factored logistic regression. And what we do is that the intermediate layer of the 2 models is forced to be the same thing. This is why the information will be shared between the two models. And so we can leverage the prior brought by the autoencoder to improve the learning of the discriminative model. So in terms of performance, we can show that the accuracy of the discriminative model is improved with this approach. We obtain systematically larger classification accuracy. Sorry, it's a bit hard to see on these images, and you don't have the full images, but also the brain patterns that are predictive of the different tasks are much improved, because if you take vanilla logistic regression, the pattern is very noisy, because logistic regression here solves an inverse problem, which is very, very hard, very high dimensional, and so the solution is very nasty. You need to regularize it in some way. But the autoencoder brings some data-driven regularization to the solution, and you can maybe see that the solution provides some blob structure, which are much more like what we expect from these brain images. And so essentially the data themselves provide a useful constraint to have a more interpretable model. Another approach to solve this scalability problem is to do compression. So I've said that the images are very large. Each image is a vector of size 200,000. So we should reduce it in some way. A standard way to reduce it would be to use random projections. That's a textbook solution for that, which works, but it will be suboptimal when your signal has structure. If there is some structure in the signal to be used, probably using it will provide a better model. That's what we do by exploiting the fact that, for instance, two neighboring voxels are very likely very correlated. So you would like to group them together. So instead of doing random projection, we propose to do some clustering where we group neighboring voxels to obtain a much simpler models, easier to solve, better conditioned, and much faster. So this is the idea. Take a bunch of training images. With the clustering algorithm, you can learn how to group the pixels, so that pixels that have correlated are in the same group. And then for any other features, you can apply the feature grouping to create a reduced representation and work on this reduced representation. So it's a lossy compression of the data. The good point is that you can reconstruct the images from this, because the reduction operator is trivially invertible. So the next step is to find a good clustering method to group voxels in the image. We have proposed an algorithm which is basically linear time in the data. So it's a recursive nearest neighbor problem, where we essentially take all pixels of an image, we associate them with a nearest neighbor. I don't know if you can see, but here it's what is written here. And then we can do that recursively again and again until you obtain a simplified representation of the same image that retains the essential contrast, just loses the high frequency details. And we can show that this is pretty good. Sorry, the result on face images is here. It's much better than, for instance, hierarchical methods that will tend to percolate. They tend to create one giant component, which is very bad in terms of data representation. Okay, I won't go through all details of the analysis, but the point is now we do a classification task on the data. So we have a classification into 20 classes of a large data set, 8,000 images. If you do the analysis on the raw data, non-reduced, you see the accuracy climbing with time. So I'm sorry the image is cut, but it takes about 30 minutes to obtain the asymptote accuracy. While with our data reduction, we can obtain the same level of accuracy in about 2 minutes. So just reducing the data and solving the reduced problem is so much faster that well, it's really worth the cost, the effort. And actually, you can't see it, but you need to believe me here. Actually, the final accuracy of the compressed model is higher than the non-reduced model. And this is a bit surprising. You don't have this effect with random projections. You know, random projections, they are very fast too, but they saturate at a certain level. While here, the local averaging of the signal has an additional benefit. It's a smoother of the data, so it increases the SNR in the data and actually boosts the classification accuracy. Okay, we have many more results on this. I see I'm a bit, it's almost one. Maybe I should skip the third part.
We are all here for you.
So it's not very long, it's 10 minutes. It's a different topic. I'm sorry, it's a bit heterogeneous with the beginning of the presentation. I think it's exciting, so I'm going to do it. I think at the time now, we are in an interesting period where there has been lots of developments in artificial intelligence, and I think that some of them can feed back to cognitive neuroscience. That's what I'm going to describe. So I'm going to focus on vision, which is, of course, easier to study system typically, because it's a large system. It has been studied also for a long time. So here in the experiment, the subject had been viewing natural imaging, so one imaging at a time, so with 3 flashes in 1 second. And they have viewed about 2,000 images, so a pretty large data set for the standards of brain imaging. And then we have the activity of their brain, the response to these 2,000 images recorded as fMRI scans. What we can do is to use a vision model, a modern vision model like a convolutional network, and analyze the input images with the ConvNet and compare the response of the ConvNet to the response in the visual cortex. And so what we do essentially is to predict the response voxel by voxel given the coefficients produced by the convolutional network. Here we used a pre-trained network, so some provided by Pierre Sermanet, a student from Yann Lequin. A network that has been trained previously to solve object recognition tasks. The nice thing with this convolutional network is that they are multi-layer. So there are, depending on the way you count them, there are about 10 layers here. And these layers represent, in an artificial vision system, different levels of complexity of the representation of the input. The first layer is essentially an edge detector in the input image. And the last layers, we know that they encode very well the categories of objects when the network has been trained to do object recognition. So what we do is that for each layer, we try and see how well the coefficients of this layer predict brain activity, the response to the 2,000 images. So more in detail, if you look at the first layers, you will see that they explain very well the activity in the medial visual region here, close to between the 2 hemispheres. So essentially what we call V1 and V2. If you take the intermediate layers, you see that they still explain V1 and V2, but they start to explain a bit more the activity in the periphery of the visual system. And the last layers explain fairly well the peripheral activities in the ventral visual cortex and the peripheric visual cortex, but not at all the activity in the medial regions. So actually what we obtain here is a mapping where you can label each brain voxel. So sorry, this big thing here is a brain, an inflated brain, which is a simpler representation of the brain activity or the brain's organization. So here you see the V1 region here and the V2. So these 2 regions are best explained by the first layer of the convolutional network. But then V3, V4 are better explained by the second layer, etc. You've got a smooth gradient between the layer of the convolutional network and the activity of the voxels. This was expected, of course, but until 4 years ago, we only had good candidate models for V1 and for these specialized regions of the temporal cortex. We knew that V1 is essentially an edge detector. I know that it's a simplification, but when you look at it with fMRI, it looks actually like an edge detector. And we also knew that these regions are responsive to object categories, but we have no mechanistic model for the intermediate regions. For instance, for V3, V4, there was no model. I'm not saying that the ConvNet is the model of the visual cortex, but at least it provides a statistically accurate model to explain the activity that occurs in these regions. So a different way to look at these results. Oh, by the way, all this was done by Michael Eichenberg, who is in the lab of Stefan Mala. You may know him because he's been at ENS for 1 year now. So another way to look at this is to look for different regions of the brain, how well they are explained by the different layers of the network. And you see that the first area, V1, V2, V3, are best explained by the first layers, while some more advanced regions are better explained by the last layers. And this holds for different subjects. So if we capture these trends, we can also compare what we obtain with different types of images. These are for discrete images that were presented, so still images. And this is for a video experiment where the subject was reviewing a video. And we see the same organization with a bit more SNR in that case. But the functional fingerprints are consistent across the two experiments. Okay, now the last thing is that with this deep network, we actually have a generative model of brain activity, because now assume any stimulus, you could look at the coefficients, the representation of this image through the convolutional network, and then predict the activity map that will follow from this stimulus. And so we've done the experiment of probing this data simulation. So we have created some artificial stimuli not seen by the subject, but for instance with this wedge pattern rotating in the visual field that so highlights different positions in the visual field. We can analyze these images through the ConvNet and then generate ConvNet activation images with the model that you have just learned, and then analyze these fake brain images and recover what we know as retinotopy, so the differential response of the visual cortex to different locations in the visual field. So, and this model, we have checked that it is the correct one. Similarly, you can take face images or place images, look at the response by the ConvNet, generate activation maps, and for instance, do the difference watching a place versus watching a face. So we know the ground truth because the subject has viewed a face and place images. We know that in this temporal occipital sulcus, there will be more response to places, while in this, I think this is the extrastriate body area, you will have some response to faces. So first we do that with some stimuli that were actually shown to the subject, but left out from the data set to do cross-validation. And actually we see that the prediction on the left-out stimuli is very close to the ground truth. So we predict fairly well the positive and negative spots in the image. And we can even do that on completely different stimuli never seen by the subject with a different background, so a gray uniform background. So we took stimuli from another experiment, simulated the response to those stimuli, and once again we can also recover the face minus place contrast, which is very similar to the actual contrast in these subjects. While if we were only using the first layer of the convolutional network, so all the contrast, the low-level contrast in the images, we don't have a good recovery of the activation maps, which can be quantified with precision recall curves, etc. It's time to conclude now. So my main messages are that for brain imaging, but for all fields, I think the field of genetics has the same constraints. we need large data sets to obtain reproducibility. And I would even say for cognitive imaging, we also need some a lot of variability, a lot of heterogeneity in the way we stimulate the subjects. What I mean is that when you present a face image, once again, the brain response can be driven by some factors that you don't model in the experiment, but are just some artifacts of your experimental setting. You want to combine the data from different labs to kind of average out these confounding aspects, hoping that the biases of different labs will cancel when you average this data. So we need some level of heterogeneity to build a brain model that reflects the true cognitive task and not some experimental artifacts. Second point is that cognitive neuroscience leads to tough multi-label problems. Tough because we have lots of labels with unbalanced problem, etc. I haven't talked a lot about technique, but these problems are quite difficult in terms of machine learning. Third point, when we go to big-ish data, since each sample is very large, then we need to really address the computational issues with online learning, subsampling, and compression approaches. And finally, I would like to suggest that current development is in artificial intelligence. So I have illustrated this in the case of vision, so the successful convolutional networks used for vision. But I think this will go in the next years to many more fields like auditory activity or the recognition of action, so more abstract representations, possibly language, certainly language actually. I think the developments in artificial intelligence in these fields will certainly feed back to cognitive neuroscience. Maybe the most important slide is that to do good science, we need good data. That's obvious. But we need also open software and, in our case, good machine learning software. So in my group, we have a very strong development effort. We have launched the scikit-learn library, which is a machine learning library in Python, which is now very popular. And we have also launched some libraries for one called NiLearn to do this task, this machine learning task on brain images. Cognitive neuroscientists, they don't master well these tools. They are complicated to use. So we try to make it as easy as possible to get images and annotations, create a problem that can be solved with machine learning, and then get some easy visualization of the results like brain maps that make sense to neuroscientists. So this is what we're trying to do, because only open science will really solve these difficult problems in the long term. With this, I would like to acknowledge the contribution of my colleagues, maybe especially Gaël Varocco, who has have a leader position in many of the works I've presented, but also Andrés Hidrobo, Arthur Mensch, Michael Eichenberg, so he's still a postdoc here, and Alexandre Abraham. And also thank the people who share the data, because data sharing is essential for the field. And thank you for your attention.
All right, we have time for a few questions.
Thank you very much for the very nice talk. I have a question about the last part of your talk, where you try to predict what the brain activity is going to be for the retinotopic mapping and the place versus face areas. I was wondering, how do you interpret the difference, the residual difference between the prediction of your convolutional neural network and the actual data from the experiments? Is that coming from the fact that you are using different images, different subjects, the network itself was trained on different things? How do you interpret the difference?
To me, the first effect here is just stimulus sampling, okay? We are working on limited data. We have a few tens of samples, represented faces and places in this data set. So we cannot sample perfectly these responses. So we are sensitive to some correlations of the data that, when you're working with small samples, you are sensitive to artifacts in the data. And artifacts are correlations. The fact that, I don't know, maybe a face, it will be more frequently represented on a light background or something like that, which can be learned by the model in some way, and is of course wrong and will impede generalization. I think it's typically the case where more data will improve this. Yeah.
I will ask some provocative question. So last year we had a series of seminars by Yann Lequin. Mm-hmm. And what struck me, they can handle much larger data. For example, at Facebook, for example, they can recognize several billions of people, faces in the photographs a day.
Yeah.
Okay. Of course, they
know everything about you. Yeah. Yeah. They know everything about you. Yeah.
So the question is, what is just the computing power? What makes the difference? How they handle so large data sets in real time? It's just for example, they have layers with more 120 layers and things like that. And what makes the difference? I mean, is because just they have a lot of computing power and storage capacity?
So there are two very important things here. So the technology, backpropagation, etc., have been done for 30 years. What makes the difference with 5 years ago was ImageNet, so a nice, well-annotated image database with 1 million samples, and this makes the difference. And second, GPU computing. So, and of course, also some technical improvements like dropout. But I think the main thing was data. So being able to assemble large data sets, and second, leveraging the computation power of, say, GPUs. In our case, I'm not using GPUs, as you've seen. I've not discussed about that, because I want this technology to be available for cognitive neuroscientists. And you cannot ask a neuroscientist to be an expert in GPU computing. And currently, you cannot use these tools well without some level of expertise. So I want most learning algorithms to work on the computation infrastructure available in laboratories, which don't have an army of engineers like Facebook has or Google has, etc. So our position, and I think it's also deeply rooted in the scikit-learn philosophy, is to empower the user who have rather standard computational architecture and still be able to run some parallel computing, so use all the CPUs that you have. And remember that the main bottlenecks for computations are our memory, typically. So try to address the memory problems in the algorithms so that we can use effectively our computation power. But I completely agree with what you said here.